Good afternoon, everyone. Is my audio clear?
Yes, sir.
Yes, good. I hope you are all doing well. So, in the last class, we have seen some examples about, or we were discussing about two important things. One of it is properties of stem cells, and in that properties, we discussed two important ones. That is self-renewal, and the second important point was differentiation or plasticity. Plasticity is the ability to differentiate into how many different types of cells, right? So in that we discussed about totipotent. That means the ability to give rise to a complete organism, which means all the different types of tissues organized in proper way, so that it is a functional organism. Similarly, we have pluripotent after that. That means it is all cell types, except that it cannot give rise to complete organism. And the example for totipotent is zygote in animal systems. We speak about that. ICM, inner cell mass. That is the example for pluripotent. And multipotent is they can give rise to many types of cells. That's one example is hematopoietic stem cell, and unipotent is hepatocytes. Right. The goal of this slide is to understand how over the time, when the zygote is dividing into ICM, is becoming an ICM type cell. ICM is giving rise to primordial germ layers, which each of them would be, say, ectoderm, mesoderm, endoderm. And each of these are again differentiating to give rise to different cell types that we have in our body right now. That means the degree of differentiation is increasing from zygote to what we have, say, hematopoietic stem cells. So the relationship between the degree of differentiation and plasticity. Plasticity is the ability to give rise to different types of cells or differentiate into different types of cells. So from as the cell keeps differentiating, the potency, the plasticity reduces. Okay, zygote is totipotent, ICM is pluripotent. From then on, you have primordial germ layers and so on, which are multipotent, oligopotent, and eventually unipotent. I think we also discussed that now, while this differentiation has happened in this direction, we have a completely formed body, and each of them have their different tissues and so on. And in all the tissues, you have stem cells, and that keeps the cell number homeostasis. What is meant by cell number homeostasis? Homeostasis is maintaining the same number. Whatever it is there, maintaining that same number, same conditions. Here we are saying cell number homeostasis. That means maintain the same cell number. Again, we also discussed about the rate of production of cells, and from external based on the usage and external damage on wearing and tearing, cell death also occurs. So there are adult stem cells that keep all the cell number constant so that the organ can function effectively within the body. So these adult stem cells keep on producing functional, highly differentiated cells, and they will perform the function, right? I hope this is a good quick review of what we have learned. We have learned about epidermal stem cells that are present in the skin that will keep on replenishing the skin, for example, or gastrointestinal tract. So these are some of the same things, and we discussed about human embryonic stem cells, and there are about 210 cell types in our body. And embryonic stem cells or even totipotent stem cells, one of the things is they can replicate unlimited number of times. Every other cell types, they have little bit of limitation. They can go divide only 50 times or so, right? There is also one more term that I think we already introduced, which is called as developmental totipotency to germ cells. So the germplasm is the only cell type in our body that can give rise to a complete organism. But there is only one point: is fertilization should have occurred. If fertilization occurred, they will give rise to a full organism. So they coined the term or phrase developmental totipotency. Have you seen this figure here before? Did we come to this slide before? Can anybody say? Okay. So this is multipotent stem cells, and I'm going to discuss about hematopoietic stem cell. These can be considered adult stem cells as well. That means multi means many. They are giving rise to many cell types. Hematopoietic stem cell will differentiate into multipotential stem cell, and then they divide into two lineages. One is the lymphoid progenitor cell. The other one is myeloid progenitor cell. Lymphoid progenitor cell makes the cells that are required for the lymph, and the myeloid progenitor cells make the cells for the blood, mostly red blood cells, platelets, to connect all the tissues in our body, all the organs in our body. Blood is the one that connects everything. It carries chemicals, oxygen, nutrition from one to a, and it also is the source how it removes the waste from our body. It brings it to the kidney or liver and detoxifies it or eliminates through urination. So these are hematopoietic stem cells are present in the bone marrow, and they keep on producing these cells. And the cell life, the life of one red blood cell, I think, is about 3 months or 90 days, and after that it dies. And this is assuming there is no external damage or additional damage. And that means we need to keep on producing. There is red blood cells are dying, or many other cells are constantly dying, and they should be remade. And they are made by these adult stem cells. Majority of them are multipotent. They can give rise to more than one type, but not all types of cells in the body. The same way you also have for stromal stem cell, which will give rise to different tissues of the different cell types of the bone. Okay. And brain stem cells also are thought to be there, but they're not as prevalent, or people have difficulty understanding or trying to know if there is a function of brain stem cells in an adult human. Say if a baby is growing from infancy to the adulthood, there is a synthesis of or production of more new cells. But the activity of brain stem cells is, some people have trouble, or there is still debate going on. But there must have been brain stem cells, because we recover from injuries, brain injuries as well. Anyway, we are learning here the general things, general concepts of how the body is organized and how stem cells keep the body alive, growing, regrowing, or regenerating. So there is one way of classification, which is based on sources. Embryonic stem cells, this is wrong, ICM. ICM is the source for embryonic stem cells. Fetal stem cells you can get from cord blood cells and so on. I think we will discuss that today. And adult stem cells are there. Usually they're never pluripotent. They're multipotent, oligopotent, or unipotent. Okay. So what we are interested in is we have been reading. This lesson is dedicated for embryonic stem cells. We now know about the adult stem cells that all the tissues or the organs have adult stem cells, and they keep on producing more based on the requirement. And then there are cord blood stem cells. We may have discussed this before, but let me try to say the umbilical cord that is present. There must be a baby here who the baby is taking the nutrition from the mother through the placenta and the umbilical cord and sending out the waste. So there is a lot of blood that is present also in the umbilical cord. When they, upon C-section or so, the umbilical cord is collected and they clip on both the sides. They put some clips so that the blood in the umbilical cord remains as such. And this is transported to the clinic where, or sometimes in the hospital itself, where the blood is drawn and some cell types, some stem cells are isolated from there. There is also, if you see the cross section of the umbilical cord, you will see that there are two arteries and one vein, and there is a lot of mesenchyme-like tissue, which is referred to as Wharton's jelly. Some people have isolated some stem cell-like cells even from Wharton jelly. Okay, so it is a good source of stem cells, and people store their umbilical cord stem cells and cord blood stem cells, you can say. And if the person is in need ever in life, they could probably take these stem cells, derive into whichever cell type they want, and put it into the person. These are all medicines of the therapeutic strategies of the future. And that is why nowadays we are having more and more stem cell clinics where they offer to save these cord blood stem cells in them and provide us whenever we need for a fee. So we will discuss about this particular another type of classification. Basically, is it's in the text in the lesson that is there. So, but otherwise, we will read in this course two important ways. In this lesson, we are already reading about human embryonic stem cells. There is one whole lesson dedicated to mesenchymal stem cells. And this is hematopoietic stem cells. It's a comparison that is given here, but we have better information than what is given here. Okay, we will discuss that. So embryonic stem cells, they have pluripotent markers like the OKSM I discussed, OCT4, KLF2, SOX2, and CMYC. And they have high expression of telomerase. We also discussed this because they can undergo innumerable number of, theoretically speaking, they can undergo cell divisions any number of times. That is possible only because if telomerase activity is very high. So that is one other marker. And these cells can be made into any of the germline cells. Okay. So they have, say is it better to use embryonic stem cells or it is better to use adult stem cells? That's the question here, right? If I want to save some of my cells for the future, it would have been nicer if I was somehow able to save embryonic stem cells. Because anytime I need anything in the future, I could probably take the embryonic stem cell, derive that particular cell type, and put it into me. If we want to save adult stem cells, then we have to save adult hematopoietic stem cells, brain stem cells, epidermal stem cells, all these things, right? That is one of the smartest way, but again, there are limitations to it. And that is where we will also discuss about pluripotency here. So why embryonic stem cell technology is much more sought is because of this. They just get one cell or one cell type in a vial of cells, and that can give rise to any other cell type. That's nice, fascinating to, or logically fitting one. But where do we get embryonic stem cells? We get from ICM. If we remove ICM, where is the person? The person is not there, right? So there is another way that is called as induced pluripotent stem cells that we will discuss in a later chapter, where this is artificial or genetic reprogramming that can be done to make normal cells as pluripotent stem cells, similar to that of embryonic stem cells. So in today's class, we are discussing about the whole scheme, the general scheme and the technical details. The general scheme is this for all stem cells. First one is to somehow get the zygote, right? You need to get the embryos. Second part is to get the derive the embryonic stem cells. That means this ICM, inner cell mass, that is present here should be put onto the plates and should be successfully be grown or subcultured. The third part of the technology would become to be able to differentiate whichever cell type we want. Here, for example, neuronal progenitor cells. If you want to differentiate them, you need special techniques. So in today's class, we will discuss mostly about how to generate the embryos. And the second one is how to derive the embryonic stem cells from blastocysts. We all know, we already discussed this, where the egg, one of the follicles in the ovary, matures and releases the egg, and the egg travels through the fallopian tube, and if there was deposition of sperm, the sperm would have traveled the opposite direction, and fertilization would have occurred in the oviduct or fallopian tube. Then the zygotic genome activation occurs. That means the metabolism of the egg, which is dormant for so long, will activate and it will start dividing. So it will form a morula, and later by about in 7 days or so, it becomes a blastocyst, and then it implants into the endometrium. From then on, it starts absorbing the food nutrition from the endometrium, and it will grow into a fetus. But what will happen is, and again on the biological side, if you notice this, if it is a menstrual cycle, if menstruation has happened here, and this one is the next one, the duration is about 28 days, and about 14th day or 12th day, somewhere around here, the egg is produced. Ovulation occurs here. Right? This is the time for preparation of the endometrium in the uterus, and this is the time. If fertilization occurs, there will not be any more menstruation. If fertilization did not occur, then menstruation would have occurred here, right? Cleaning up the endometrium, and a new cycle of menstruation starts. So about three days or so, or 2 days maybe, the egg is actually open for fertilization. So when we talk about this complex mechanism of things, there are so many problems that may arise. First thing is the follicle maturation. I hope you have heard about PCOD or PCOS, which is it's a complex problem with hormones or lack of activity, diet, and many things could influence it, or maybe genetics, I don't know. Is where not one follicle matures, but all of them try to mature, because of which none of them actually mature. Right? A follicle, only one follicle should have matured, and then that will burst and release the egg out of the ovary. So kind of ovulation problems are there. First, in the female side, we are talking about ovulation issues that are there. There could also be another issues where there are no eggs at all. No eggs. Because there could be, assume there is a disease, or there is a kind of modification or problem because of which eggs have not formed. We will study about a lesson or so, maybe, where we will discuss a little more of it. And there could be other problems where the egg should be able to move from here and into the uterus, and then implant upon fertilization, if it has fertilized. So the traveling, if the oviduct is crooked or something like this, say, then the egg may have, or this zygote may have problems moving through it easily. And in some cases, there are also something called as ectopic pregnancy, where the blastocyst starts developing here itself into an embryo, and that is, the embryo will die anyway because there is insufficient supply of food, but it may also cause a problem to the mother. So those should be terminated whenever possible. And those are some of the problems. There is also another set of problems from the male. The sperm count is one of the problems. What if the individual, that is the partner, is not having enough of sperms? You should have billions of sperms, so that one can increase the probability of some of them moving or traveling up the oviduct and meeting the egg and fertilizing it. So sperm count is one serious problem. The second one is the motility of say if the sperm count is fine, but the motility of them is weak, then too they cannot swim up. And that is the other problem. So these days, if you see, there are in vitro fertilization clinics in even in an average town. Previously, it used to be one in a country or 3 or 4. Nowadays, you see it everywhere, which is only indicating that it's good business. Why is that good business? Because there is a lot of demand. Why is there demand? That means there are many people who are having trouble with conceiving children. There is also one more issue with the females, is that the uterus, although everything may have happened, but they may not be able to hold on to the embryo. The miscarriage may happen, right? The mother goes through a lot of these things, a lot of problems associated with it. So what do they do in vitro fertilization? Is they will hormonally stimulate the female to produce superovulate, or to ovulate, have many eggs, and say they will retrieve the eggs from the ovary, and they will take the sperm from the man, and they will mix them here. And in vitro is for vitreous or glass, right? Or test tube baby, you call them. In vitro fertilization, we are adding we are facilitating the fertilization to occur in the petri plate or test tube, and then take the embryo and implant into the uterus of the mother. So that is basically the simplest way of saying in vitro fertilization. So what if, say for example, they have taken one egg, there are problems with this couple, and that's why they are at the in vitro fertilization clinic. So they have taken one egg from here, from the female, and then fertilization occurred, and they put it into the uterus. For some reason, the egg did not grow properly, or there is miscarriage. Then what to do? Again, we have to go for another round of isolating the eggs, and then in vitro fertilization, and so on. So what the technicians usually do, or the doctors, they collect more than one. They collect multiple eggs, many eggs, and they perform the in vitro fertilization of all of them. And they take 1 or 2 eggs and implant it here. If it fails, they already have eggs. Those eggs, that the other eggs will be put it as frozen. They will be frozen and kept aside, stored. So if the pregnancy works out, well and good. If it doesn't work out, they will take from the frozen ones and they will implant in the new way. So the mother doesn't have to go through all the cycles over and over. Okay, that's how the in vitro fertilization clinics usually work. So here is what to do with those extra, these frozen eggs. Say the couple is happy and they left, and we have frozen eggs. What to do with them? Here we need to also try to think of in terms of ethics and empathy. If you type a title something like on YouTube, if you type "The Heart of a Lioness," there was a show on National Geographic or somewhere long time ago, which is where the lioness, a lioness, was has caught a newly born gazelle and did not eat it, did not hurt it, did not let it go far away from the lion, and did not let any predator try to kill the young gazelle, the newly born gazelle. It is as if it has adopted the young gazelle. And why would that happen is usually it happens when the lioness has lost her cubs. If it is a young mother and newly born cubs were killed or lost, the mother instinct is always present. Say this is a pair of instincts. How it happens is a mother and child, newly born child. A mother has instincts of caring for something and giving lactation and giving milk. The child has an instinct to cry until it receives the milk, it receives the care, it receives the touch, it receives the attention. And this is a perfect couple of instinct, right? It is a reciprocity of instincts. And that is the nature of, and over a period of time, when they keep on sharing, a bond between the mother and child happens. Okay. Now, think of a typical woman in India or in any conservative society who is not able to conceive, right? Say, first thing is, it might not be her mistake at all. It could be the man that is the problem. But this is typically, if you talk in, at least in the rural conditions, it's a male-dominated society. The woman who didn't conceive will have a problem, because it's not just that first thing is she is not able to live her mother instinct. One of the problems is that psychologically. Second thing is the society also looks down on her. The third thing is there should be a purpose for all of our lives, right? Usually, if our reproduction is the objective of life, and then the next purpose of life after reproduction, why are we alive? And there the next is to find purpose, to raise the children, to nurture the children, raise them, make them better, and be able to survive longer and happier, right? So there are purpose problems, instinct problems, and the social problems. Social problems even is the worst of them, because whether she has or she doesn't have, it is nobody's business. And people still have this kind of notion: she doesn't have any, she couldn't have any. There are name callings you might have seen in movies and all those things, right? People may not say it openly, but there are issues, at least under the blanket, that we don't know. So people talk about it bad. So how can these people find purpose? How can these people find to get to live their motherly instincts? And that is where the embryos, they can actually probably adopt a child who is not well and adopt a person. If that person is 24 years old, is there any attachment? Is there anything at all to the person who is adopting? That's not so much. If it is 10 years old, probably there is something and there could be some attachment. What if the child is just 1 year? There is more give and take between them, mother and the child, and then the bond is stronger and longer. What if they were able to donate, they were able to get the embryo, and then they give birth to the child of their adopted child? Then they will be able to see that the purpose of the mother or the parent will become to give rise to or give birth and raise the child as if it is their own. Everything except the genes are theirs. At least emotionally and psychologically, they would feel they have lived their instincts to the completion. So this is where you can give birth to your adopted child is if the parents have agreed, they can probably donate their frozen embryos to some childless couple, and they might be able to give birth to the child and then raise them as if they're their own. They are their own. I think we should say that. But otherwise, what do we do with those surplus embryos? We donate it. That is one of the best way of doing stuff. Disposal is not so good, and using for donating for research, it is a nice way of doing for future. But I don't know how many people would want their embryos to be cut open. I mean, embryos, we are talking about blastocyst, okay, not at a fetus level also. So, and some of them they keep them frozen for longer use. Maybe something happens, we will take at a later time. So what the parents do with their frozen embryos, it is their business. They can choose what to do. Again, this is an ethical perspective, right? It's a topic of ethics. What if we should say, no, they have to always give it to childless couples? We cannot be the judge of what they do with what belongs to them. We cannot say the same thing about they should give it to research instead of disposing. We cannot say that. These are sentiments and sensitivities that are involved. Some people will feel that the embryos that have come from me should not be broken, tortured, cultured in tubes. They have religious or any kind of situations they may have or conditions. So this is how we think about those things. But why we are talking about this is the only source of human embryonic stem cells, if anybody has donated to it. You cannot isolate an embryo from a living human like this. It's very difficult, tedious, and may not be fruitful because the egg is small and you have to go through operation on many things, which is very difficult. So the only way, legal way, is to get these frozen embryos of the couples, and you should always get informed consent. That means the parent should have signed. They should know what kind of research is going to happen on those embryos, and they should have agreed to it. Only then we are allowed to use them, but otherwise we cannot use them. Right? They can file a case if we have used and did not reveal what we are going to do with the embryos. Okay, that is meant by informed consent. In many clinical trials and other things, in many research papers which involves humans, we'll ask whether the people who participated in the experiment, not the researchers, but they are people who are donating the materials or tissue cells, they were informed. Did they agree to the research to be carried out or not? That is called as informed consent. So the next step is we have now, say, from the frozen embryos, we got the embryos. We should now be able to isolate embryonic stem cells. This is going to get a little bit like a technical session from here on. Just try to see what you can. So what is ICM? What are embryonic stem cells? They're just, you take the ICM, put it and culture it in the medium. They are embryonic stem cells. There is no big difference. It is inner cell mass as long as it is in the blastocyst. Once you take it out and grow them outside, then they are embryonic stem cells. So there are three ways. One is called the whole embryo method. The second one is called as immunosurgery. The third one is called as mechanical suppression of the ICM. These three will remain the same. I don't know if you are learning immunology now, I guess you are learning now. So I will go a little bit brief and I will repeat in the next class so that your understanding will be good. First one, the whole embryo method. If you see the blastocyst under a microscope, it looks something like this. I mean, it's three-dimensional confocal. You'll see a layer like this. So the outside layer you're seeing this is the zona pellucida, and this part is the ICM, inner cell mass, and the outside big cells you see are the trophoectodermal cells. These cells are trophoblast cells, you can say. So first thing is we have to remove the zona pellucida. To remove the zona pellucida, we use an enzyme called as pronase, which will digest the zona pellucida, and now we have a naked embryo, naked blastocyst. Remember that we are trying to take, the ICM is what we need to grow it as embryonic stem cells, but it is within a structure called the blastocyst. Normally, what would happen in the uterus? In the uterus, there is the lining of the uterus, and at the blastocyst stage or so, it implants into the endometrium and start absorbing nutrition from the outside. So we can try to mimic such scenario by providing a layer of cells. That is referred to as feeder layer. The function of the feeder layer, as in the case with the uterus, is to provide some biochemical factors, provide some attachment sites, and things like that, to create a kind of environment where the cells would grow as they should be. So this feeder layer are usually made from either mouse embryos or mouse fibroblasts or human fibroblasts. And they are treated, if these are fibroblasts and you add the medium into it, they might also divide in and form more number of cells. We don't want them to divide. We are looking to actually only multiply the ICM. All right. So there is a way to arrest growth of the feeder layer cells by using mitomycin C or irradiation. That is, somehow you will prevent cell division to occur. Cell division occurrence will require that the DNA is intact and so that only then the cells will go from G1 phase to S phase to start with. So if you're irradiating or using mitomycin C, you are stopping cell division further. So there is a feeder layer which serves sites of attachment, produces biochemicals and other signal molecules which may be useful to the growing cells, but they themselves will not divide because they are arrested using mitomycin C or irradiation. So the medium would include several components. This is the basic one that many of them use. This is called as Delbecco Minimal Eagle Medium. And this is HES for human embryonic stem cell medium. Different labs have different compositions. They may have additions and so on. The protocols may vary a little bit. And there is also fetal calf serum and fibroblast growth factors. So the medium constitutes all these things. They will have amino acids and many other nutrition in that. These are names for those recipes in a way. So if you leave that, we call this the whole embryo method because we are putting the whole embryo onto into the Petri plate here. And then in about 2 weeks or so, you will have two different types of cells. Trophoectodermal cells would have grown into large cells like this, and inner cell mass would grow into a lump like this one, right? These are taken and subcultured, ensuring that these cells do not come into the new plates. And now we have an established embryonic stem cell line. So how to identify these two is that there is a difference between them. One of these, I discussed this before, nucleoplasm to cytoplasm ratio. right? The embryonic stem cells or the pluripotent stem cells usually have high nucleoplasm to cytoplasm ratio, whereas the trophoectodermal cells represented here, they have low nucleoplasm to cytoplasm ratio. That means the ICM cells have less cytoplasm and trophoectodermal cells have large cytoplasm. Okay, this is whole embryo method. You have to remember pronase, you have to remember the feeder layer, how it is there and how it is inactivated or arrested in cell cycle. And the other one is medium, and then the morphological differences between how ICM grows and T cells grow, and then subculture. The next method is called as immunosurgery. Immuno is using immunocomponents, something like antibodies or complement system. Surgery is you're trying to cut open using the immunocomponents, right? So I like this method a lot because it sounds nice to see how these components can be used to achieve a target. The first step will remain the similar, that you treat with pronase to get rid of zona pellucida. Say here, I will just erase this and put anti-human. So assume now you have a naked blastocyst. Okay, the blastocyst is ball of cells with blastocele and ICM, inner cell mass. So inner cell mass is not exposed to the outside, only the whole ball is there. So you add anti-human antibodies. That means these antibodies are raised against humans. I hope you know from your 10th class or +2 and so on, antibodies look something like this with their proteins, something like this. They have 2 light chains and 2 heavy chains. This region is where through which it interacts with antigens. Antigen is usually the foreign part or so. And they may have some structures like this. These are referred to as epitopes. If you already are studying immunology, you might have come across this already. I'm unsure. So using these antibodies are usually bivalent. They have through this structure, they interact with the epitopes. And here you have the heavy chain parts with constant regions. So the antibodies will bind to the blastocyst because they were raised against the anti-human, say for example here, like that. And then in the next step, they add guinea pig complement system. Complement system is a series of or cascade of proteins which will activate one after another, and they will be activated by this the constant regions or this region of the antibody. When they come in contact with it, they get activated, and then they cause the cell lysis. So you have added anti-human antibodies, and then you added complement system of guinea pig, and that would have lysed all the trophoectoderm cells. All of them would have been lysed. Now we just wash it off, and what is remaining is ICM. We take this ICM and subculture it. That is immunosurgery method. We are using antibodies and complement system to get rid of the trophoectoderm cells, and that is how we will obtain the ICM. Okay. This will be the last one, and I will stop here. So the next one method is mechanical dissection. So if you know about IVF clinics, they all have what you call it as a micromanipulator. You see here, this is the place where they manipulate the embryos. They will allow, if fertilization should occur or anything also. The same things are also used in stem cell technology to isolate any of those or manipulate the embryos. In this course, as we progress, we will see several places where we will manipulate some the embryos as well. So what they do is they have a suction pipette. I will try to get the videos in the next class. They have a pipette for suction, and this is an embryo. And if you can recognize it, it only has about 1, 2, 3, 4, 5, 6 or so. So this is in morula stage. Morula stage is where cell division has occurred. It's a pair of cells, and it is completely, they're all identical. This part of it is zona pellucida, right? The outer thick covering. So they use two pipettes. They are very tiny, so they cannot handle with hands. They have very fine glass pipettes, which they use suction to hold this pipette here, to hold the embryo like this. And then they use needles to make an opening through the zona pellucida and reach the ICM and take out the ICM. And then go for subculturing. That is the mechanical dissection of embryos. I'll stop here, and then if you have any questions, I would like to take that. Any questions? Anybody?
It's clear, sir.
Okay. Well, you're all free to always ask any questions anytime. You can also WhatsApp or email. Google Classroom would be a good place to post your questions so others can try to answer as well. Ritika, did you find out the information that I asked?
Yes, sir. It was the stem cell bank life cell. It's actually in Chennai, sir.
Yes. Then?
They're just preserving it, sir. No, other services, we are not sure, sir. We're just preserving the stem cells.
I know. Price?
Price, my answer, it was about 1 lakh when they...
It's a one-time payment?
Yes, sir.
Okay. Yeah, good. Okay. How long they would have said, for 20 years or something, or you don't know?
No, sir. No idea about it.
Okay. Okay, then. Thank you very much.
Thank
We'll
you.
meet in the next class. Take care and have fun.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.